Hi everybody, we're going to start the chapter on blood. If you have your course pack handy, let's turn to page 22. Blood is actually an unusual thing for us to spend a chapter on because it's a tissue, not an organ system. You might recall that blood is a connective tissue, and of course that's why we talk about it, because it's a liquid connective tissue. The liquid portion of blood is around 55% of the blood's volume. We call this liquid portion plasma. Around 45% of the blood is formed elements. That's just the name we give to those blood cells, like red blood cells, white blood cells, and platelets that are found in the blood. In a human body, the blood constitutes around 5 to 6 liters or quarts in a male body, and around 4 to 5 liters or quarts in a female's body. So because we have a variation here due to body size, testosterone, muscle content, we actually say just on average there's around 5 liters of blood in the human body. That way we don't have to distinguish between males and females. To put this in perspective, these are 2-liter bottles at the bottom of this page. There's 6 of them down there. So an average person has 2.5 2-liter bottles of blood in their circulation. Just an interesting thing to note that you can go into hypovolemic shock, meaning that you have lost 10 to 20% of your whole blood volume. That's not a whole lot of blood to lose when you're considering that you only have around 5 liters to begin with. In terms of blood characteristics, blood is slightly basic or alkaline with a pH of 7.35 to 7.45. The temperature of blood is slightly warmer than body temperature at 100.4 degrees Fahrenheit. Interesting to think about that for just a moment. Blood circulation helps to keep our bodies warm. Imagine if blood was colder than average body temperature, it would actually cool you down instead of maintain your warmth. Blood is also thicker than water. The term for thickness is viscosity, so we can also say that blood is more viscous than water. Blood is also bright red when oxygenated. That's because when oxygen binds iron molecules on hemoglobin, it creates a bright red color. Blood is dark red when deoxygenated. That means that the oxygen molecules are not bound to the iron molecules on hemoglobin. We oftentimes say that, blood is blue when it's going through veins, but truly it's never blue in our bodies. The blue color that we may see in some of our vessels through the skin is actually a trick of the eye. It's a combination of the melanin in your skin that provides skin pigmentation and the way that your eye perceives color. So there's not really ever a time where your skin does appear, or your blood rather appears blue. Let's talk for a moment about blood functions. Blood transports gases like oxygen and carbon dioxide. It also transports waste products to the kidneys, for instance, for removal from the body. Nutrients and hormones are also transported through the bloodstream. Blood also helps us to regulate or maintain a body temperature. On average, that's around 97-98 degrees Fahrenheit. Blood also is important for its pH. We know that the pH of blood is somewhere between 7.35 and 7.45, and it is critical that it stays within that very narrow range. We've already learned about how blood and water content have to be regulated. Thinking back to the endocrine system, can you tell me 2 hormones that influence the water concentration in the bloodstream? One is antidiuretic hormone, or ADH, and another, aldosterone, in which water passively follows sodium back into the bloodstream. Finally, blood plays a role in protection. It protects us from fluid loss through its clotting mechanism, in which blood turns from a liquid to a gel-like state, and blood also protects us from foreign invaders. Those white blood cells attack and remove those foreign invaders like viruses and bacteria from our bodies. So let's talk about what's in a drop of blood. This takes us down to the very bottom of page 22 in your course pack. Like we talked, a little more than half of your blood is liquid plasma. That plasma constitutes on average around 55% of whole blood. The formed elements we know are the blood cells, red blood cells, white blood cells, and cell fragments known as platelets. They constitute around 45% of the whole blood volume. Plasma, when separated from the blood cells, takes on a somewhat straw or yellow colored appearance. In this centrifuged blood here, you can see that the plasma is floating toward the top with that straw or yellow colored appearance, and the blood cells sink to the bottom because they're heavier. In fact, the blood cells, red blood cells, make up approximately 45% of that whole blood volume. Only around 1% makes up that buffy layer right through there, and that is composed of those white blood cells and platelets. The remaining 55% or so of blood is the plasma. And so we know blood plasma contains largely water, solutes, amino acids, nutrients, hormones, and waste products. Sometimes people do need a plasma donation, and in that case, we'll centrifuge away the plasma, may return those blood cells back to the donor, and then the recipient can get just the blood plasma. So sometimes burn patients, for instance, might need the plasma and not those red blood cells. You'll notice in your course pack that around 90% of your plasma is water. So that is the primary component of blood plasma. I should mention to you some of these plasma proteins because they are very important, and they'll be part of our discussion throughout this course. We make plasma proteins in the liver, such as albumins. These are very important for maintaining a type of blood pressure that we call blood osmotic pressure. So you've probably heard of osmosis, right? That is a process by which water is attracted to solutes. Well, water is also attracted to proteins like albumin. We'll talk more about albumin's role in maintaining blood osmotic pressure a little later on. We also have plasma proteins such as globulins. They could be antibodies or thyroglobulin that transport thyroxine through the bloodstream. Or we have clotting proteins like fibrinogen that are found in the blood plasma. Let's turn over to page 23 and now talk about the living formed elements in your bloodstream. These are your red blood cells, platelets, and white blood cells. You'll want to know that we are listing them here from most common down to the least common. In this picture, you've got a blood smear in which the blood plasma has evaporated off, so you're not really seeing that blood plasma. Instead, you're seeing the blood cells. We know that the most common type of blood cell is what? Erythrocytes. Those are red blood cells, or RBCs for short. You can recognize them easily because they have no nucleus. In fact, in this picture, there's lots of red blood cells because they make up the majority of what we find in the formed elements. The second most common type of cell is actually not a cell at all. It's instead a fragment, and these little tiny things here are the platelets. All right, and so if you look, they look like just little pieces of matter in the background, but there's several here for us to take a look at. If you were to count, you would notice that in this particular picture, they are the second most common type of formed element. The third most common type of formed element is the white blood cell. So these are very easy to spot because they have a nucleus that will pick up some type of stain or dye. So here you actually have two white blood cells in this image. Let's talk first about the erythrocytes or red blood cells. We know the prefix erythro means red. And of course, cyte means cell, so these are our red cells. These have one function only, and that is to circulate in your bloodstream transporting blood gases, primarily oxygen, although some carbon dioxide will be transported by these red blood cells too. Red blood cells make their ATP anaerobically through glycolysis. Let's talk about this word anaerobically for just a moment. Anaerobic means without what? Without oxygen. So glycolysis is a way for us to make ATP, that's the goal there, by breaking down glucose directly into that product. There is no need for oxygen to help us transform that glucose into ATP, since red blood cells do this anaerobically. Now, why are we talking about this? Why is this important to our discussion of the function of red blood cells? Well, look at what they transport. The primary product they transport is oxygen, and of course, if they are burning through that oxygen to make ATP, they'll arrive at their destination with no supply. So I kind of imagine it like this. If you're driving a donut delivery truck, but you eat all your donuts, by the time you arrive at the grocery store with your donut supply, You open the back and there's nothing left, all gone. So it wouldn't make very good sense for the red blood cells to utilize all of their oxygen before they made it to their destination. Let's talk about the anatomy of erythrocytes. They are shaped like biconcave discs, meaning they look like two frisbees placed back to back. So biconcave means concave on either side, just like this. So if we were to turn these red blood cells in this picture on their side, they would have indentations on either side like you see here. They're also somewhat flexible like a frisbee because they have to squeeze together to get through the small capillaries of our bodies. And then you can also see they lack a nucleus because there is no dark staining nucleus visible in these cells. They also lack most organelles. They are simply for one purpose only, and that is to do what? They have one job, and that is to transport blood gases like oxygen and some CO2 around the capillaries. Once they're worn out, because they have no nucleus, they are not repaired. They're simply removed from circulation and new ones are being replaced constantly. Let's talk about this term here. The hematocrit is a measure of the proportion of the blood volume occupied by red blood cells. Well, we know the most common type of formed element in your bloodstream is a red blood cell. It just happens that the majority of your formed elements are those red blood cells. In fact, we said that 55% of your blood was what? Plasma. About 1% or so was something called a buffy coat. And around 45% of our blood are those red blood cells. And of course, that means that that's a pretty big volume in your whole blood volume, 45%. The range here is about 48% for males to around 38% for females. The difference has to do with testosterone driving higher levels of red blood cell production in males. So we can simply look at a centrifuged column and measure the hematocrit. In this picture, you'll notice that the hematocrit is equal to the number of erythrocytes. They're both 45%. Now, if the buffy coat makes up less than 1%, that means that the plasma is approximately 55% in a typical volume of blood. This, of course, has been centrifuged. The reason we know this is because blood cells are heavy and they will sink to the bottom. The plasma is lighter and it will float toward the top of this centrifuged column of blood. Red blood cells get their color from the hemoglobin molecules that bind and carry oxygen. So let's talk further about a typical hemoglobin molecule. Hemoglobins are made up of two different kinds of proteins. They are alpha chains and beta chains here. And each of these chains has inside of them a heme group. There's one there, there's another, and each heme group has an iron molecule sitting on it. So if you count, there are a total of 4 chains sitting here. There are 4 heme groups. There are 4 iron molecules. And guess how many oxygens we can transport? We can transport 4 oxygen molecules. So keep in mind your magic number of 4 for the hemoglobin molecule. All right, so this just summarizes for us what we talked about. In fact, hemoglobin is a quaternarily structured protein. Quaternary, again, means 4th level. That is the most complicated structure for a protein. And 2 types of proteins make up those hemoglobins. And of course, the polypeptide chains are globin chains, and the heme groups as well. They're also protein-based. When oxygen binds hemoglobin, it forms what we call oxyhemoglobin. It is bright red in color, and we abbreviate oxyhemoglobin as oxy-HB. When oxygen unbinds from hemoglobin, it forms a dark red color. We call that deoxyhemoglobin. Now, we talked about that issue with blue before. There's never a time when your blood is truly blue, but we sense that color through our skin thanks to melanin and the trick of the eye of perception. Did you know that carbon dioxide can also bind to hemoglobin, but it does not compete for a seat on the iron molecules? Instead, it binds to the amino acids, therefore there's no competition for a ride. So just interesting to note here that mostly we're transporting oxygen, but we can transport a small amount of carbon dioxide here. All right, so oxygen is bound and it forms oxyhemoglobin. When oxygen unbinds, it forms deoxyhemoglobin. Just interesting to note that the hemoglobin molecule shifts in shape in order to make it easier for more oxygens to bind. By the way, what was the max number of oxygens that can bind to a hemoglobin molecule? Magic number 4. If you'll turn the page with me, let's head on over to page 24 and talk about erythrocytes and the count in blood. Different textbooks will give you different numbers here, but they're more or less in a similar range. In a single drop of blood, and that's a cubic millimeter, that's a single drop of blood, we're going to find between 4 and 6 million red blood cells. If you do the math and you think about you've got 5 total liters of blood, 45% of that is red blood cells. The number is a staggering amount of red blood cells in circulation at any given time. Red blood cells tend to live around 120 days. They don't live very long because they have no nucleus. So why is that important? Well, the nucleus gives us information to do repairs. So without a nucleus, this cell can't be repaired. It simply goes to the liver and spleen to be recycled. Speaking of recycling, red blood cells are snared in the spongy tissue of the spleen, or if you don't have a spleen, the liver takes up that job. When red blood cells travel through, they become trapped. So healthy red blood cells are allowed to continue on in circulation. It's the damaged ones or worn out ones that are trapped and removed by the spleen or liver. We don't actually waste a lot here. We use iron molecules to make new red blood cells. Iron can be stored in your liver. The heme is broken down to become bile. Keep in mind, heme itself is a protein. It breaks down to form bile pigments. Bile ends up going into the digestive system, and it helps to emulsify fats. So we don't waste anything here. Eventually, bile is removed either in your feces, and that's why your feces are colored brown, or if it's broken down into circulation, it is also removed in your urine, and that's why urine appears yellow. So, interesting thing to note here, the color of your blood comes from the hemoglobin molecule. The yellow color of your urine comes from the breakdown of heme, as well as feces, the brown color there comes from the breakdown of bile. So we've got a lot of stuff here to take a look at. Globin chains can be reused to make new proteins. We can recycle those amino acids and make any kind of protein from their building blocks. Let's talk about making new red blood cells. This is a process we call hematopoesis. That's a fancy word that simply means growing new blood cells. Now, where does this take place? This occurs in the red marrow of bones. Keep in mind from Bio 210 you may have learned that there is both yellow marrow and red marrow. Red marrow is the only one involved in making new blood cells like white blood cells, red blood cells, and cell fragments called platelets. This word here, hemocytoblast, breaks down to mean blood, cell, and blast is a cell that forms. So this is kind of like the great-grandfather or grandmother to all of our blood cells. This is a precursor stem cell that can give rise to any and all of our formed elements. Red blood cells, white blood cells, and platelets all differentiate from that hemocytoblast. Cool word there. Here you can see the hemocytoblast up top here. It does give rise to all of these types of formed elements. You'll notice that red blood cells follow this pathway over here on the far left. And over here on the far right, we're looking at the pathway that platelets use. And here in the middle, we actually have white blood cells and how they are formed from hemocytoblast. So if you have problems with your red bone marrow and those hemocytoblasts, look how it influences the production of all formed elements. Let's talk about making red blood cells specifically. This process is driven by a hormone called erythropoietin. EPO for short. Of course, that erythro prefix means red. Okay, so the stimulus for this process begins with the kidneys. And of course, the kidneys monitor blood pressure for aldosterone's release. We are now going to learn that the kidneys also monitor oxygen levels. When the kidneys detect low levels of oxygen, they release this hormone called EPO. We know that EPO, like any other hormone, travels via the bloodstream. When EPO arrives at the bone marrow will increase red blood cell production. Okay, so here's your stimulus, low oxygen levels in the kidneys, and the output is increased red blood cell production. That's our goal. The process of making erythrocytes can take around 3 to 7 days. Different textbooks will tell you different things. Some will tell you it may take as many as 2 weeks to make a new red blood cell. But we tend to see the process run from these hemocytoblasts, eventually resulting in an anucleate red blood cell. What do we mean by an anucleate red blood cell? This word here. That means no nucleus. So these precursor cells here that lead up to producing these reticulocytes and erythrocytes actually have nuclei. But once those cells are developed properly, the nucleus is ejected or kicked out, and what is put into circulation is a reticulocyte. So I'm going to draw a line right here because now we're going to put those reticulocytes into blood circulation. Give those reticulocytes some hours and they will eventually mature to the point of being able to transport oxygen. And at that point, when they're fully functional, they are now called erythrocytes. So kind of think of reticulocytes as blood cells with their training wheels on. They're not quite there yet, but once they kick off those training wheels, they're a full-fledged erythrocyte able to transport blood gases. So if we want to know your potential, your future potential for transporting oxygen, why do we look at your reticulocyte level and not at your erythrocyte level? Well, your erythrocytes are being recycled every minute of every day, but your reticulocytes are brand new. They are not being recycled because they are the up-and-coming population of your erythrocytes. In fact, they should live for, on average, 120 days. In order to make red blood cells, we need a source of iron, which can be stored in your liver. We need vitamin B12. We'll talk more about that with the digestive system. And we also need another B vitamin called folic acid. So think of these as the ingredients needed to make a red blood cell. By the way, I'm going to pause here for just a minute. Take a time to fill out these blanks on page 24 of your course pack. All right, it's a good time to pause and think back now. What stimulates the release of erythropoietin or EPO? It is letter C, blood oxygen levels. Low blood oxygen will stimulate EPO. Hey, just as a side note, EPO is used in blood doping in athletics, not a good thing. And of course, this gives somebody a competitive advantage because it increases their red blood cell population. And if you increase your red blood cell population, you increase the amount of oxygen you can transport and the amount of ATP you can make. And that can increase the amount of muscle activity and your athleticism. So that's why it has been used in the biking and cycling industry as a way to increase the amount of energy a person has in that particular field. Let's talk for a moment about leukocytes. They are found over on page 25 in your course pack. These are the white blood cells of our body, and they fight infections and form antibodies. Now, they're a very specialized group of cells. They're not one size fits all here. We're going to talk about the various types of leukocytes and how they fight infections in our bodies. Now, these particular cells have nuclei, organelles, and perhaps maybe have granules. They can travel in and out of blood vessels through a process we call diapedesis. Literally, that word means walk across or walk through. And that's important because we want to have these cells reach the site where there may be an infection. White blood cells are the only ones that travel by diapedesis. Red blood cells and platelets don't have this particular feature. Notice the normal blood cell count here is much lower than a red blood cell count, only 6,000 to 9,000 per cubic millimeter. By the way, this will vary depending on your book. So, if you have a book that has a slightly different number, this is tweaked based on lab values. Leukocytes can live from hours to days, or even memory cells can live for years. So there's, again, not one size fits all here for these leukocytes. We can separate the leukocytes into two categories, those with granules and those without. We'll call those with granules the granulocytes. And we'll call a category that has no granules the agranulocytes. Of course, that A prefix there means without. So in this picture, it's pretty easy to see the ones on the right, the fills are the granulocytes. And the ones on the left here that have very few granules or none at all, they are called the agranulocytes. So I don't mean to imply that the agranulocytes have no granules whatsoever. They just tend to have less than you find in a granulocyte. So this is a great picture for us to take a look at for just a moment. All 5 types of leukocytes shown here do have nuclei. That is easy to spot because it is a dark central portion in each of these cells. And these little tiny things in the background, those are called the granules. And I also want you to take a moment to compare sizes here. These agranulocytes on the left, especially that huge monocyte, tend to be larger than our granulocytes. Finally, one last point to make in these pictures. Take a look at the word fill. Okay, so our granulocytes have a suffix of fill. These particular cells love a stain color. Fill means to love. So neutrophils love a neutral stain. Okay, basophils love a blue stain, and eosinophils love a red stain. Monocytes and lymphocytes, well, they're not really lovers and they don't particularly care for a particular color of stain, so we're not going to see that in the case of those two cells. So we're going to target the chart on your worksheet and talk about granulocytes first, and then we'll talk about the agranulocytes. All right, so here we know about the granulocytes, the fills. They do have nuclei that are lobe-shaped. Okay, they are larger than red blood cells, but they're not quite as big as those agranulocytes. And most of these are phagocytes, meaning it is their job to do what? They eat. Okay, look for the granules in the cytoplasm. That's something else that's good to look for. So here, let's take a look at a typical granulocyte. You can see plentiful granules in this picture here. That is something that's very easy to spot in one of these granulocytes. We'll talk more about basophils in just a moment. All right, so we've got neutrophils, eosinophils, and basophils. Let's tackle the neutrophils first. Neutrophils are characterized by a lobed nucleus. In this picture here, you're looking at the neutrophil. And it is characterized by having a 3-lobed nucleus. I'm going to label those 3 lobes. It's pretty easy to see the lobes here because they are not drowned out completely by the granules. They typically are staining a light pink to a light blue color in the cytoplasm. So the granules hopefully will not be blocking your view of those 3-lobed nuclei. Now, what do neutrophils do for a living? They are phagocytes. So it is their job to eat bacteria and dead cells. All right, so let's take a look at the next one. Another neutrophil here. Notice it's larger than a red blood cell. And interesting to note that if you happen to have an infection that you can't kick, sometimes a complete blood count will give us an idea of what's going on. An elevated neutrophil count will indicate to us there may be a bacterial infection, since that's what neutrophils tend to eat. Here's a picture of a neutrophil compared to an eosinophil. Notice the eosinophil, I'm going to circle it right now, down here, has a somewhat set of red, pink, or orange colored granules. This time the nucleus is bilobed shaped. There is one portion here and another portion there, and that bilobed nucleus is characteristic of an eosinophil, while a trilobed nucleus, or 3 lobes, is characteristic of a neutrophil. You'll notice that eosinophils tend to fight allergies and parasitic worms. Nowadays, you might imagine that those eosinophils may get less business than they did in the past. We tend to lead a cleaner lifestyle nowadays than we did maybe 100 or 200 years ago. So eosinophils may have less to do in terms of fighting those parasitic worms, but more to do in terms of fighting those allergies. Beautiful eosinophil here. Notice those lovely red colored granules in the background and that bilobed nucleus. Basophils are blue in color, great purplish blue colored granules here. Lots and lots of granules. In fact, so many that it's very hard to see the nucleus. If you were able to see the nucleus, which is kind of hidden back here, you would notice that it's bi or trilobed, meaning how many? 2 or 3 lobes. Now, these are the most rare of all of our red white blood cells. In fact, this one releases histamine. Histamine is involved in an inflammatory reaction. You know, when you have an itchy mosquito bite, that itchiness is partly due to the histamine reaction that comes with inflammation. Histamine happens to be a vasodilator. That means that it will cause your vessels to relax. Okay, so basophils actually have the potential to kill us, unfortunately, because histamine reactions can cause anaphylactic shock. If you're stung by a bee and you're highly allergic, if you eat shrimp and you're highly allergic, you can cause a massive drop in blood pressure thanks to these cells here releasing their histamine. So what a rare cell can still lead to death. All right, so we just talked about the phil group. Which one of these will likely lead to which one of these will a bacterial infection likely lead to a high level of? That is neutrophils. Agranulocytes fall into the category of being lymphocytes or monocytes. There may be a few granules here, but for the most part there are none. Notice the lymphocyte is rather large in size. This particular cell here helps to form antibodies. Okay, so if you get in contact with the virus, this cell makes antibodies, so that if you get in contact with it again, you will hopefully be able to fight it off quicker the second time. Lymphocytes are matured in your thymus and bone marrow. You may have heard of T cells and B cells before. That's because they're named for their location where they're matured. Monocytes are another type of agranulocyte. They are rather large cells with a kidney bean-shaped nucleus. In fact, I think that looks like a Pac-Man. So that nucleus has a very distinctive shape. Notice there are no granules in that cytoplasm. Okay, so this particular cell also happens to be a phagocyte, which means it does what for a living? It's a big eater. Now, this particular cell can migrate out into your cells and tissues and find infections that it can then eat. So it is not confined to travel in the bloodstream. It can migrate through diapedesis where it needs to go. So in summary, here's your 5 types of leukocytes. You may have heard of the trick to remembering these. Never let monkeys eat bananas. Never are neutrophils. Let are lymphocytes. Monkeys are monocytes. Eosinophils and basophils. This is from most common to least common of our white blood cells. So a great picture here that gives you a complete set of all those cells to take a look at. I would review this section before you go on further to make sure you're comfortable with identifying these different types of white blood cells. You'll also want to know what each type of blood cell is responsible for doing. For instance, which type fights allergies or parasitic worms? That's an eosinophil. Which types can be phagocytes? There's 2 answers here. Monocytes and neutrophils. Which type can form antibodies? That would be a lymphocyte, for instance. All right, so there's our mnemonic device one more time. Never let monkeys eat bananas. Great way to keep these straight from most to least numerous. And now let's wrap this page up by talking about platelets at the very bottom. We know platelets are also known as thrombocytes, although they're not true cells. In fact, these are cell fragments formed from the rupture of a very large multinucleate cell called a megakaryocyte. We know mega means large, kary means nucleus, and of course a cyte is a cell. So these particular big megakaryocyte cells are kind of like balloons that have been blown up just too much, and they rupture off these little tiny fragments that we call platelets. Those platelets are great for clotting blood, but they don't last very long. Without a nucleus or really any organelles, they only live around 10 days. You'll want to make note of that in your worksheet along with the normal blood cell count, which is around 150,000 to 350,000 per cubic millimeter. That is, per drop of blood. So here's a look at a megakaryocyte. This huge cell here in the center is that megakaryocyte, and it constantly leaches off little platelets like this that enter blood circulation from the red bone marrow. So that's where we are. We're in the red bone marrow with this megakaryocyte. And in this picture, you can see the tiny platelets here in the background in comparison to those red blood cells and very few white blood cells there. Kind of cool picture here. You can compare the red blood cell on the left, this one here. Of course, that's a white blood cell, and one guess what that is, right? That is a platelet.